We are going to discuss zonulin and intestinal permeability. Is there a connection between this protein and changes in permeability of the gut? We're going to be doing this in two parts, and the first part is dealing with pathophysiology, inflammatory response, zonulin and its effect on intercellular tight junctions, and the increased intestinal permeability, which is an outgrowth of that response. We know that there are many factors: stress, various toxins, food particles, many different drugs and pathogens, and even organ malfunction that can cause gastrointestinal inflammation. And that in turn can lead to various types of food intolerances, leading on to changes in the immune system, with an endpoint being autoimmunity problems. What I'm going to do right now is concentrate for a few moments on gastrointestinal inflammation. And we know that inflammatory response underlies virtually all degenerative conditions. As you can see here, Alzheimer's disease, coronary artery disease, diabetes, and so on. And it is the integrity of the gut lining which is now understood to play a fundamental role in determining the degree by which inflammation is able to affect the human body experience. Now, what about the intestine? Well, we know that it is the largest surface of the body. In fact, it is estimated that it takes up to an area of about 2 tennis courts. That's quite large. And within the lumen of the gut is a microbiome, about 130 trillion bacteria. And to give one an understanding of what that means relatively, we have about 10 trillion cells in our human body. So this is 13 times as many bacteria as we have human cells in the body. And they are continuously exposed to the environment by bacterial, dietary antigens, and lying very close to the cells of the gut is the largest accumulation of immune cells in the body. In fact, 70% or more of all lymphoid tissue in the body sits intimately attached to the cells of the gut. What is zonulin? Well, zonulin, or to be more particular, the zonula occludens toxin, or ZOT, was discovered a good number of years ago. It is an enterotoxin that is produced by Vibrio cholerae, and it was discovered that it regulates the intestinal tight junctions, the intercellular junctions between enterocytes in the gut. Now, we have in recent years been able to determine that on chromosome 16 there is a pre-haptoglobin 2 gene, which is in fact the human analog of this Vibrio cholerae enterotoxin. And it regulates fluid movement, large molecule and immune cell movement between body compartments. And I think one can begin to see that this could be an extremely important protein that regulates intestinal movement. This is a diagram that comes from Fasano, who did incredible work with his team out of Maryland. This comes from a 2011 paper. And really what you see here, if you look at the top left area, you see that between two enterocytes, the two cells that line the gut, there in fact is a very, very strong closure of the intercellular space, normally not open at all. This is what's called the tight junction. And one can see that if certain antigens or fragments of antigens end up coming into the gut and are able to interact with some of the receptors at the surface, in fact, the CXCR3 receptor in particular, you then get a cascade of response which is D88 dependent, will cause the stimulation of zonulin creation. That zonulin, that protein, then leaves the cell and is able to actually activate some of the receptors on the enterocyte surface within the gut lumen, specifically PAR2 and EGF. And when the zonulin is able to interact with these receptors, the result of that is a decoupling of the proteins which are part of the tight junction, allowing them to open. Now, if they open, you then can get the movement of antigens which normally would not get into the body. They are then able to move freely into the body cavity. If they do, you then get a tremendous response, an antibody response, both IgA and IgG. Remember that IgA represents the largest amount of mucosal immunoglobulins, and IgG the vast percentage of immunoglobulins in general. So with that antibody response, you then get the movement of cells throughout the body, which causes the types of immune reactions which we're going to be discussing in a few moments. One can inactivate zonulin by, in fact, closing off those receptors, and there is some wonderful work that's taking place in the last several years with something that is called larazotide, which is now going through clinical trials, and if in fact is able to come out as a medication, will be able to be used to not permit zonulin to have its effect of opening up the intercellular tight junctions. If you want to take a look at the actual expression of these CXCR3 receptors, here is another one of the slides from that 2011 paper in which you see the red area on the left on the surface of the cells. This is a human small intestinal biopsy, and you can see that relative to the controls, there are a lot more CXCR3 receptors. The gene expression for that, which you can see on the bottom, shows you that in active celiac disease, that's what the CD stands for, you have much greater gene expression than you have with the controls. And when CD, or when celiac disease goes into remission, it enters very close to the control level. So clearly these receptors are extremely important. The intercellular tight junction itself is quite complex and consists of a whole series of proteins with clodine and occludin, the two proteins that create a very strong mesh in the intercellular space, and they connect to other proteins, to ZOT1, 2, and 3, as well as actomycin filaments, which together create a very, very strong bond, keeping the intercellular space very, very tight. Here's another picture of that, showing you the tight junction with these various proteins, which therefore normally would not allow the passage of macromolecules. However, when you do have various types of situations that could cause them to open, we now understand that what was formerly considered as a static system, tight junctions which don't change, we now know that they are quite dynamic and that they adapt to both developmental, physiological, pathological, and of course, many dietary circumstances. And what they really do is they will control the passive absorption of nutrients. They are able to create a very strong surveillance of the gut microbial content, and as we said earlier, that is a vast microbiome. They're also able to defend against some infectious agent, which is one of the reasons why this was found as a cholera enterotoxin, because in fact, what the body was trying to do was to get rid of the cholera bacterium by really flushing it out. And of course, it is important in the transport of white cells, specifically leukocytes, into the gut. when you actually look at some of these tight junctions, there's some very, very beautiful work that was done by Fasano, which shows you, for example, on the top, the resting state, in which these proteins form a very tight mesh, whereas when the pathway is activated, you can see the opening taking place, and this is electron microscopy, so that you can see that this would allow the passage of macromolecules. The other thing that is important to note is, as we said before, there is a tremendously large mucosal immune system, specifically the gut-associated lymphoid tissue, also known as GALT, and that is the series of immune cells which lie in intimate contact to the enterocyte and the lamina propria. These cells lie very close and are ready to be able to interact very quickly with the epithelial cells in order to handle non-self antigens, antigens which are coming from outside the body, as well as the vast microbiota in the lumen. So the zonulin effect on GI permeability has now been shown to be quite dynamic. For example, here you see a work by Tripathi et al., and you see that both in small intestinal permeability and gastroduodenal permeability, the gastrointestinal permeability percentage change between 3 days before and on the challenge day with zonulin or with a regular haptoglobin shows you that you need the pre-haptoglobin, the zonulin, to be able to actually create a very significant change in permeability. The most interesting thing about this, however, is that it's reversible. And that is that if you were to look at it 48 hours after that zonulin stimulation, what you find is that you have gone to a permeability which is back to its control levels. So zonulin effect on permeability is reversible. So here is a bit of a busy slide in which we basically then tell you that if you look at intestinal hyperpermeability, what is in parlance called leaky gut syndrome, but really an intestinal hyperpermeability or increased permeability, if you disrupt the gut lining due to stress or antibiotics, etc., food intolerances, you can get, if you go to the right, improper absorption. And IgG antibodies are produced. If you go to the left side, you'll see that this is true with multiple food intolerances where IgG antibodies are produced due to gut inflammation. This gut inflammation can also be caused by IgG antibody food antigen complexes which are actually deposited in the gut. And the effect is an inflammation that worsens with improper absorption. And of course, that would cause even more IgG. So you have almost a feedback loop that takes place. Now, what health conditions do create increased intestinal permeability or leaky gut syndrome? The autoimmune disorders, we know about them, the multiple food and chemical sensitivities, chronic fatigue syndrome, heart failure. We know this now, but depression, chronic inflammatory conditions, which we will go into in much more detail soon, chronic pain, and of course, chronic yeast overgrowth syndromes. The common imbalances that we are quite familiar with are malabsorption or malnutrition, the effect of bacterial or yeast overgrowth, and with that, immune activation because of an impaired integrity of the gut, a degeneration of the intestinal lining for many reasons, a dysbiosis, which is extremely important when one wants to deal with diets and supplements, and of course, the whole realm of food sensitivities. And what you get in terms of symptoms is a spectrum that goes from severity, both from mild right through to severe. You get bloating, poor digestion, all the various signs of inflammation, the food sensitivities, the chronic pain, what is known as brain fog or poor memory, depression, and of course fatigue, both acute and chronic. And so over the next few moments, what I'd like to do is to just go through a very, very general description of the causes of increased intestinal permeability, also known in parlance as leaky gut. But we're going to call it increased intestinal permeability. And it runs the realm, as you see here, of many different factors. We're going to begin by looking at diet itself. Now, we do know that diet can promote increased intestinal permeability. We're a fast food nation. Excess sugar, which is almost epidemic, the effect of gluten, casein in milk, the whole realm of processed foods, and of course alcohol. And in fact, the standard American diet and alcohol is now very well known to lead to a risk of developing this increased intestinal permeability or leaky gut. So what are food sensitivities? Well, there are three categories classically of diet-induced inflammation. The first is a true food allergy, which affects about 4 to 6% of the population. Autoimmune response, which is about 3 to 5% of the population. And then the really vast amount of food sensitivities, which affects 30 or 40% or even greater of the population. And that leads, food sensitivity does, to non-allergic or non-IgE response. That is the same as you would consider with non-celiac gluten sensitivity, which needs to be seen differently from classic celiac disease response. It's 6 to 8 times as prevalent and stimulates both the innate and the adaptive immunity response. Immune system, in fact, has two arms. The innate or natural or native response is immediate or within hours. It is non-specific, and it is activated by antigen chemical properties. This needs to be seen very differently from adaptive or acquired immune response that is not immediate. It can take days or weeks. It is antigen specific. So that is auto-regulated. That's what gives us the memory of immune response throughout our life and a future immune response. So how do food sensitivities cause inflammation? Well, we do know that there are triggering mechanisms. They are necessary. It's got to be food antigens, chemicals like haptens, various pharmacological products, immune complexes, either IgG, A, M, and the lectins. When you do that, you get a cellular activation, and the cellular activation are all the lymphocytic cells, the eosins or basophils, neutrophils, monocytes. All of those can be activated, which then leads to mediator release. And this is key because the mediators, these proteins, the cytokines, histamine, the various prostaglandins, and others, are really what then are responsible for the pathophysiological effect, for the clinical or subclinical inflammatory response, which we all know well: the increased gut permeability, the various types of tissue damage, the pain receptor activation, which will come forth, the smooth muscle contraction, and of course the neurological and endocrine responses. And you then have this tremendous number of effects, which we are not going to go into in detail, but clearly one can see that it runs the gamut from depression right through to arthritic diseases, ulcerative colitis, Crohn's, etc. What about medications, and what kind of effect does that have on increased intestinal permeability? Well, we do know that corticosteroids can have a very profound effect, as can antibiotic substances, antacids, of course, and the xenobiotics. This tremendous number of substances which can have a very devastating effect on intestinal permeability, like the dioxins and the polychlorinates. In the case of infection, there's some that we know well. There's no doubt that intestinal viruses and bacterial overgrowth can have a very, very strong effect. Yeast overgrowth, extremely well known to have an effect. all the parasitic infections, and Helicobacter pylori, which we know can not only cause ulcers, but is now implicated in many other gut problems. When we talk about stress and its effect on intestinal permeability, one can see that you have what's called a stress-induced hyperpermeability cycle. Very, very large phrase, but basically it begins with an increase in stress. That causes a hypersecretion of cortisol by the adrenals, which then leads to digestion and absorption problems, can be quite compromised, which then leads to intestinal mucosal inflammation of various degrees. That in turn leads to increased production of cortisol, which of course then can have even more stress-induced issues. So you can see that this can become a very, very difficult cycle to break. Hormonal imbalances in general have an extremely important effect on the increased hyperpermeability. The hormonal imbalances are from progesterone, testosterone, estrogen, the thyroid, and the deficiencies of any of these can lead to significant changes in intestinal permeability. For example, if you look at estrogen, estradiol is able to modulate the paracellular permeability and the tight junction function in the intestinal epithelium. Testosterone, or a lack of testosterone, delays intestinal healing, and we know that treatment with progesterone is able to protect the intestinal lining from various types of macroscopic injury, albumin extravasation. And finally, thyroid, or a lack of thyroid stimulation of both the gastric and intestinal cells, can lead to significant ulceration and intestinal permeability, which of course is quite often known as the leaky gut syndrome. And finally, autoimmunity. We do know that increased intestinal permeability plays a very large pathogenic role in various autoimmune diseases. And it is the loss of intestinal barrier function which is necessary to develop autoimmunity. And this came from the Annals of the New York Academy of Science, and it has now been seen in many, many studies. And so our next part is actually going to deal with the pathogenesis of the autoimmune and systemic diseases.